In the name of Allah, today we are going to discuss about nucleic acids. First of all, has to say that it's molecule, an organic molecule that store information for cellular growth and reproduction. Growth and reproduction which is happening inside of the cell. And nucleic acid is a kind of polymer made up of nucleotides. It means nucleotides are monomers for the nucleic acid. It means lots of nucleotides joined with each other and made nucleic acid chain. Nucleic acid is composed of three units, which is phosphate group, sugar group, and base, which is nitrogenous base. So the first group, phosphate group, is always phosphoric acid. The second group, which is sugar group, is pentose sugar or 5-carbonate sugar, that we have two kinds of pentose sugar, deoxyribose and ribose. This is the main structures of them. The difference between deoxyribose and ribose is having hydroxyl or hydrogen group. The third group is base, nitrogen base. We have two sort of it, purine and pyrimidine. If it's pyrimidine, means it's made up of only one ring. And examples are thymine, uracil and cytosine. If it's purines, it means there is two rings. It means this nitrogen base structure got two rings. For example, adenine and guanine. So, about nucleic acid, if you remember, we said lots of nucleotides combined and made nucleic acid. So, lots of structures made up of phosphate group, nitrogen base and pentose sugar combined with each other and made nucleic acid. So if we talk about functions of nucleic acid, we can say that it passes heredity characteristics from one generation to next and to trigger the manufacture of specific proteins. It means helping to carry on the process of synthesizing or creating or producing specific proteins inside of the cell. What types of nucleic acid we can say? There is two sort of nucleic acid. First one is DNA or deoxyribonucleic acid, which is located in nucleus of a cell, mitochondria and plastid. The second form is ribonucleic acid or RNA, which is also in nucleus, ribosome and cytoplasm, freely swimming on cytoplasm surface. Now let's talk about DNA. DNA or deoxyribonucleic acid is a kind of nucleic acid which is responsible for storing genetic information and passing this information from one generation to another one. It is made up of nucleotides, by the way, and also located in nucleus, mitochondria and plastid, and information inside DNA, genetic information, is getting stored as codes, and guanine, cytosine, thymine and adenine are bases which are responsible for storing the codes. About DNA, we can say that it is a double strand molecule, means it's made up of two strands, as you can see, and it's a polynucleotide structure, meaning it's made up of lots of nucleotides. And okay, as you can see that these two nucleotides are running in opposite direction from each other. Two strands are running in opposite direction. The internal part is bases. It means nucleotides are joining with each other with bases. Bases are connecting with each other, and as you see, adenine base makes 2 hydrogen bonds with the thymine base, and guanine base 3 hydrogen bonds with the cytosine base. It means the bonds which are connecting nucleotides are hydrogen bonds. Between adenine and thymine there is 2 hydrogen bonds. Between guanine and cytosine there is 3 hydrogen bonds. And the other, we can say, nucleotide parts are located or running in opposite direction to each other. And these two strains, this strain and this strain, is specific, or we can say identical, and if we see only once, by seeing only one strand, we can guess what is in next part or in another strand. Adenine is always pairing with thymine, guanine is always pairing with cytosine. It means adenine cannot pair with cytosine and guanine cannot pair with thymine. Always they are pairing with each other, and this is making this strand of the DNA. About functions of DNA, we can say that storing genetic information from one generation to another one, and regulating all the metabolic activities of a cell. like making proteins. Okay, the second kind of nucleic acid is RNA. RNA is a single strand structure. As you can see, there is only one strand of it, and it's also made up of nucleotides, and sugar base and phosphate group is present in its structure, and the sugar of RNA is ribose. From the DNA sugar was deoxyribose, but the RNA sugar is ribose. Containing the nitrogen base uracil instead of thymine. If you remember, we said that adenine, guanine, cytosine and thymine was the basis of the DNA, but adenine, guanine, cytosine and uracil, instead of thymine, there is always uracil in structure of RNA, and RNA is always single strand structure. About types of RNA, we can say there is three types of RNA: messenger RNA, transport RNA and ribosomal RNA. Suppose this is the cell structure. This part is the nucleus, this part is cytoplasm. These structures are amino acids, which is the monomer of the protein. These structures are ribosome. Ribosome is the factory of cell which is responsible for making proteins. So the main function of RNA is, girls, to produce protein. So now we are going to see that how these RNAs are working with each other and make protein. First of all, mRNA or messenger RNA, which is inside the nucleus, is unzipping the DNA, placing themselves inside DNA, getting copies of one strand, getting the codes of making protein, learning from the DNA how to make proteins. Okay, then messenger RNA can come out from the nucleus to the cytoplasm and start giving that information to ribosome. Then, in other hand, transfer RNA is carrying all amino acids, which is the monomer for producing protein, and carries that all amino acids to the ribosome. rRNA is ribosomal RNA, which is involved in the structure of ribosome. So now we say that mRNA carried the codes, transfer RNA carried the amino acids. Now inside the ribosome there is codes or recipe for making the protein, and amino acids, the monomer structures as well. Now ribosome starts to make different sort of proteins, which is essential for our body. So we can say about types of RNA. Again, there's three types of RNA: messenger RNA, ribosomal RNA and transfer RNA. The messenger RNA or mRNA function is to carry a copy of the instruction from the nucleus to other parts of the cell. Ribosomal RNA or rRNA makes up the structure of ribosomes, and transfer RNA or tRNA transfers amino acids, monomer of protein, to the ribosomes to be assembled. So there is some difference between DNA and RNA. Let's talk about this. The DNA is found in nucleus, mitochondria and plastid, as you know, and the RNA is found in nucleus and cytoplasm. DNA sugar is deoxyribose, the RNA sugar is ribose. The DNA nitrogen bases are adenine, guanine, cytosine and thymine. The RNA nitrogen base are adenine, guanine, cytosine and uracil. The DNA function is to store genetic information and control the metabolic activities. The RNA function is only to synthesize or produce protein. The DNA structure is double helix or double stranded, and the RNA structure is single stranded or single helix. The DNA replicates or can copy itself by help of DNA polymerase, which is an enzyme, and RNA can replicate or copy itself with the help of RNA polymerase. It means enzymes are different, and the DNA is catabolized by enzyme DNase, and the RNA can catabolize by enzyme RNase. Now check yourself.